Every human being alive today began as a single cell. Before that, one half of that cell was already racing against millions of identical rivals through a body built in almost every way to stop it. How does one cell survive a journey designed to kill it? Why does the body destroy nearly all of its own reproductive cells before they even reach their target? And what actually determines which one, out of hundreds of millions, gets to succeed? Today, we're following that journey from beginning to end, from the moment of release to the final seconds before one cell becomes two. A single human sperm cell is about 50 microns long, roughly a 20th of a millimeter, with a head containing tightly packed genetic material and a tail built for one purpose: propulsion. A typical ejaculation releases between 100 and 300 million of these cells. Laid head to tail, they would stretch for kilometers. Of that number, only a few hundred will ever come near an egg. Only one will fertilize it. The journey begins in an environment that is immediately hostile. The vagina has a naturally low pH, acidic enough to kill sperm within minutes under normal conditions. Semen itself provides a brief defense, containing alkaline fluids that temporarily neutralize this acidity, giving sperm a short window to move before the environment turns against them again. The next barrier is the cervix. For most of the month, it is sealed by thick, dense mucus that blocks almost everything from passing through, including sperm. But in the days surrounding ovulation, rising estrogen changes this mucus into a thinner, more permeable form, sometimes called fertile mucus, opening a temporary channel that did not exist days before and will not exist days later. This is not incidental. It is the same narrow window that defines fertility itself, and it explains why timing matters as much as biology. Once inside, sperm split into two rough strategies. Some are swept forward quickly by muscular contractions in the uterus, reaching the fallopian tubes in as little as 15 to 45 minutes. These early arrivals are often the least prepared for what comes next. The majority take a slower path, covering only 2 to 3 millimeters per hour, resting temporarily in small folds of the cervical canal called crypts. Sperm can survive there for up to 5 days, waiting for ovulation before continuing their climb. Throughout the uterus, the body is actively working against them. White blood cells treat sperm the way they treat any foreign material, engulfing and destroying the vast majority before they ever leave the uterine cavity. This is not a passive filter. Some research suggests it is a selective one. Sperm that begin moving with unusually vigorous, whip-like tail motion appear to trigger this immune response themselves, drawing in the very cells that will destroy them, and effectively clearing weaker competitors out of their own path in the process. It is a strange kind of sacrifice built into the system itself. Of the hundreds of millions that started the journey, only a few hundred reach the fallopian tubes at all. Inside the tube, the surviving sperm gather in a narrow region called the isthmus and loosely attach to the tube's walls. Here, they undergo a transformation called capacitation. Cholesterol and coating proteins are stripped from the surface of the sperm's head, a chemical change that primes it for what comes later. Without capacitation, a sperm cell cannot fertilize an egg, no matter how far it has traveled. A related discovery complicates the image of sperm as isolated competitors. In thick fluid resembling the female reproductive tract, some human sperm have been observed physically joining together at the head, forming small, swimming clusters. These groups move over 50% faster than sperm swimming alone. Notably, the sperm that form these teams tend to have far less DNA damage than those left swimming individually, suggesting this cooperation isn't random, but a behavior shown mainly by the healthiest cells. The partnership doesn't last. Once capacitation is complete, the clusters break apart and each sperm continues alone for the final stretch. That final stretch also involves hyperactivation, a shift in movement where the tail begins whipping with far more force and less coordination. It looks less efficient, but it is exactly what's needed to physically push through the layers still surrounding the egg. The egg, meanwhile, is not a passive target. Cells surrounding it release chemical signals, chemoattractants, that create a trail for nearby sperm to follow. There is even evidence of selectivity in this process. Fluid surrounding an egg has been shown, in laboratory studies, to attract sperm from some men more strongly than others. This has led some researchers to describe it as a form of cryptic female choice, the possibility that the egg's own chemistry plays a role in which sperm ultimately reaches it, beyond simple speed or numbers. Fertilization itself typically occurs in the ampulla, the upper third of the fallopian tube, about 1 to 2 days after intercourse. Sperm remain capable of fertilizing an egg for up to roughly 80 hours inside the female body. After that window closes without success, they do not survive. The egg's outer boundary, the Zona pellucida, still has to be breached. Multiple sperm attach at once and release enzymes that gradually break this layer down. This is typically not the work of one cell alone. Many sperm weaken the barrier together, and only one is left to pass through it. The instant one sperm's head fuses with the egg's membrane, the egg reacts within seconds, hardening its surface and releasing granules that seal it against any further entry. From that moment, no other sperm can succeed. The race for all remaining competitors is already over. Several beliefs about this process circulate widely, and the evidence behind them varies. The idea that most sperm die along the way is true, though not primarily by exhaustion. Much of the elimination is active, carried out by the immune system itself. The idea that speed alone determines the winner does not hold up. Some of the fastest sperm to arrive are not capacitated and cannot fertilize anything. And the notion that the egg simply waits is outdated. Current research suggests it plays an active and possibly selective role in the outcome. From release to fertilization, a sperm cell survives an acidic environment, a sealed cervix, an active immune response, and a chemical gauntlet at the egg itself, usually within about 2 days, occasionally stretched across 5. Out of hundreds of millions that begin this journey together, only one is left standing at the end of it. Every person alive today is the result of that one cell succeeding, once, against odds that were never in its favor. Thank you for watching. New hope, new life.